Hello everyone, welcome to BPI Digital Week, brought to you by the producers of the face-to-face bioprocessing international conferences, visiting Boston this year and Kyoto, Amsterdam and Santa Clara next year. My name is Catherine and I'll be your host for today's session titled Research to Realisation, an insight into turning a plan into a product. First, I'll cover some quick housekeeping items. If you experience difficulties with audio or advancing slides, refresh your screen with F5. If you are experiencing other issues, hit the question mark button to receive assistance. At any time during the presentation, submit your questions into the Q&A window on the left-hand side of your screen. In 24 hours, you'll receive a link to watch the recording of this session. Let's now begin by introducing our speaker from Prometic Bioseparations, Buzz Lobezo, Product Support and Development Manager. For the question and answer portion, we have two guest speakers with us, Chloe Booth, DSP Manager, and Chris Sadler, Chemistry Manager. Thank you for joining us today, Buzz, Chloe, and Chris. Now I'll hand it over to you to begin with the presentation.
Hello, my name is Buzz Lobezo, and I'm the Product Support and Development Manager at Prometic Bioseparations Ltd. I've been with the company for the last 14 years, and in that time, I've carried out downstream processing, troubleshooting, and working with our products and customers alike. In this presentation, I'd like to talk to you about turning research into realization, whereby we can take a requirement of the customer and fulfill their unmet needs. But why? With improvements in technology, people are living longer, and with an ever-growing and aging population, the demand for new, more cost-effective and tailored therapies is increasing. This is materializing in the form of revamping legacy products where the old good enough is no longer good enough, biosimilars which require cost-effective purification solutions, improved and new biotherapeutics needing an unmet purification solution, and emerging gene therapies which require personalized products. Biotech and biotherapeutic companies are being driven to making wholesale improvements to their products and processes, including the purification steps involved. To keep up with these demands, Prometic has developed a range of technologies and strategies to complement our core expertise of affinity adsorbents. We have the tools for full process solution, whether you're looking for a novel ligand from initial screening, require support with process optimization, custom adsorbents, or off-the-shelf products. The following presentation will describe each aspect from initial concept through ligand design, process optimization, tech transfer to final product, whether it's an off-the-shelf or custom adsorbents to an open platform pre-packed GMP-ready column to support and enable our customers to deliver. The first step is to identify a ligand specific to the target and library screening. Here's the decision process for ligand discovery. The starting question is always the same. Is the target protein structure known and available? If this information is available and there is a known binding site, we can use ligand docking and virtual high throughput screening technologies. Ligand docking means that we take selected ligands from our virtual library and virtually place them in the binding site and evaluate the fit of the ligand with the binding site. Alternatively, if the binding site is not known, we will use the binding site determination or blind docking to identify ligands suitable for the target protein. This is where the virtual ligand library is screened in silico against the protein to identify ligands that have an affinity for the protein binding site. When we don't have structural information about the protein, we can use information about known inhibitors and search for ligands with structural similarities. Of course, there are also occasions when there's no structural information available or known inhibitors to the target protein. The ligand discovery approach then is via the systematic high-throughput screening of a selection of libraries, or the identification of a hit through a more discrete but diverse library. Ligand design has many similarities to drug discovery and development. This includes the identification of a collection of compounds, primary screening, identifying lead compounds, and structure-activity relationship studies. Further characterization, screening, and design of the target ligand is subsequently carried out. This then leads to secondary analyses to end up with a suitable ligand. There are, however, additional constraints when trying to identify a ligand suitable for affinity chromatography. These include the requirement for the ligand to be attached to a large solid inert support and still have the access for the ligand to the target molecule binding site. Another important requirement is that once the ligand is immobilized to the bed support, it must be in an orientation that allows it to interact with the protein binding site. Additionally, there's the balance between affinity and avidity. We want the molecule to bind selectively, but not too tightly, so as to allow desorption with relatively simple solutions or additives and without detrimentally affecting the target protein. Protein ligands and affimers, which are small proteins or polypeptides, can be effective affinity ligands for binding target molecules, but have limited chemical stability, especially to caustic, thus limiting their cleaning and reusability. Triazine ligands, which is the basis of most our adsorbents, are ligands which are small chemical constructs developed on a core triazine ring attached via a spacer arm to an agarose bead. The ligand is built up by coupling a primary amine to the triazine core, creating an intermediate. This monochlorotriazine intermediate is then further substituted to a final two-dimensional ligand. Prometic standardly creates an 8x8 array library of subtly different ligands. This building of ligand libraries using a wide range of different chemical compounds allows for the creation of an extensive library incorporating a wide variety of properties and are caustic stable. Here is an example of the primary data that can be expected from a screening. This is a representation of 4 libraries which were screened using the same feedstock against the same target protein, and the ligands were classified as being either functionally active or inactive, or whether there's non-specific binding. This can be further identified in subsequent screening or wash studies. Further on in this presentation, I will show more detailed heat maps in which measures the degrees of interaction between the library and the target or its impurities. As an overview of our approach to ligand screening, this slide summarizes when the ligand design approach is via diverse or general ligand libraries. It can take several iterations of ligand screening and design to identify a suitable ligand. Once the libraries are selected, they are screened and the data is analyzed. This information is then used to evaluate the structural activity relationship of how well the selected ligand binds to the target protein. This information is then fed into the design of the next library. For example, if the primary screening indicates that the ligand, which shows binding to the protein, is hydrophobic in nature, then the next library will be designed with ligands that are hydrophobic or display hydrophobic regions. This library is then screened, the data analyzed, and the structure-activity relationships is evaluated again. This iterative approach enables suitable ligands to be identified from a diverse library of ligand structures. In this first case study, in collaboration with Pfizer, a ligand was required as a platform for the binding of a transferrin scaffold. In order to increase the circulatory half-life of bioactive molecules, larger ubiquitous fusion partners are used. Here, transferrin, which is an 80 kilodalton glycoprotein, has been used as the fusion partner to a GLP-1 mimic, which was to be used for the treatment of type 2 diabetes. This is planned to extend the half-life from minutes to greater than 3 hours. Because we had a known binding site for the protein, the identification of the interactions between the ligands and the binding site was established by means of docking studies and virtual high-throughput screening. This was achieved by exploring the chemical space of the potentially suitable ligands using a virtual ligand library and allowing the ligands, via docking, to roam over the surface of the molecule to see how the ligands interacted with the binding site. From the virtual screening, ligands were selected and a physical library was made and screened against the transferrin fusion protein. Primary screening was undertaken and the results focused initially on the binding of the target to the ligands. In this study, 29 promising ligands were initially identified, which demonstrated a binding capacity where greater than 80% of the target was bound. Further screening identified suitability of candidates by the recovery of bound target as well as the purity of the target. The measure of a successful candidate was greater than 60% recovery with a greater than 80% purity, obviously along with the greater than 80% binding. For the screening, it was also important to ensure that the amount of residual binding of the material still bound to the adsorbent, i.e. levels of protein still in the CIP, was also low. The ligands that were identified from the high throughput screening were further evaluated in silico by docking the ligands into the binding site to see how well the fit was, and to further eliminate ligands that would be unlikely to make a good affinity chromatography adsorbent. Essentially, we did not want the ligands which bound to be bound either too tightly or not tight enough. This was determined by calculating the binding free energy to give us an indication of how well the ligand would both bind to the target protein and also if it could be eluted. Chromatography of the lead candidates was performed. The analysis of the elution fractions of the lead candidates demonstrated binding capacities of up to 14 mg per mil and recovery of high purity material, which was approximately 95% purity as determined by HPLC. There was also a strong correlation observed between the free energy of binding, determined by SAR, and the binding capacity of the lead candidates, whereby we saw a proportional increase in the binding capacity as the binding free energy increased. This is one example of a rational ligand design and screening process which PBL carry out to identify a suitable ligand for binding a target molecule. We have identified, however, that the process of setting up relationships with clients can be time-consuming as well as expensive. So to circumvent this and offer the power of diverse screening as a more accessible and cost-effective option, we, PBL, have come up with exactly that, a diverse library of ligands from across our libraries which can allow for the rapid identification of a hit, which can bind your target protein or impurities without the requirement of any feedstock adjustment. The multi-mode mimetic ligand library, or MMM plate, is organized into zones with ligands having broadly similar properties clustered together to simplify selection of binding and elution conditions. The zones, as seen here, 1 through 6, are comprised of ligands with a range of functional groups, including ionic, which are in zone 1, hydrophobic, which are in zone 6, or combinations thereof in zones 2 through 5. The MMM plates are comprised of 96 discrete packed columns with the top and bottom frits and reservoirs above. The columns will freely drain under gravity, so automated liquid handling systems are not required, but could be used if available. Otherwise, a multi-channel pipette is all that's required. The top and bottom seals need to be removed and the adsorbents equilibrated. The sample solution is then applied to all the columns, and the flow-through, wash, and elution fractions are collected by drainage into deep well collection plates. Collected fractions can be analyzed using discrete analytical tools such as STS-Page or by a variety of plate-based assays to measure the amount of target protein and contaminants in each fraction. This allows for rapid identification of ligands which bind the target but not the contaminants, as required for capture applications, or ligands which bind contaminants but not the target, as required for flow-through polishing applications. Here's an example of the use of the MMM plate, where CHO feedstock containing a monoclonal antibody, which was filtered but not adjusted, was loaded onto each column of the library, which was pre-equilibrated with phosphate buffer, matching the pH and the conductivity of the feedstock. After loading, all of the columns were washed with equilibration buffer to remove any non-bound material, and the elution was carried out with a low pH sodium citrate buffer. Analysis of the elution fractions showed a broad spectrum of binding ranging from 0 to almost complete binding of the loaded antibody. Zone 2, in particular, contained ligands which demonstrated a diverse array of functionality, from high antibody binding as well as ligands which exhibited little or no antibody binding. SDS page analysis of the eluted fractions from Zone 2 reveals some ligands bound significant amounts of host cell protein and product-related impurities. In particular, ligands C3, C4, C5, and D3 through D6, which bound large amounts of HCP, but displayed little affinity for the whole antibody. Consequently, these ligands were of interest for the development of a flow-through polishing step for host cell protein and product-related impurity reduction step for this antibody. Analysis of the elution fractions for host cell protein using time-resolved fluorescence assay, or HTRF, reveals several ligands bound significant quantities of CHO host cell proteins, particularly ligands in rows C and D. This confirmed the SDS page findings. The results are displayed as a heat map where 0 is no binding, up through 5, which represents very high binding. Comparison of the results for the elution fractions using different detection assays reveals 3 ligands exhibiting low antibody binding but very high binding of HCP. Whilst these ligands, C2, C3, and C9, highlighted in yellow, bind significant amounts of host cell protein, they exhibit little binding of product-related impurities. However, other ligands, such as A11, B11, and D9, highlighted in green, also demonstrated very low binding of the whole antibody, medium to high binding of host cell proteins, but also high binding of the product-related impurities. A summary comparison of the SDS page and HCP results for the 6 selected ligands is shown here. All 6 ligands display very low binding of the full chain antibody, but good binding of host cell protein and light chain, which makes these ligands very well suited for use in flow-through polishing applications for monoclonal antibodies expressed in CHO cells. Consequently, by screening the multimode mimetic ligand library, it is possible to identify and select multimode ligands for the binding and removal of specific problem impurities from the whole antibody without the need to adjust the applied feedstock. Here's another example where the MMM plate was screened for ligands capable of removing factor 11 from a feedstock. Now, factor 11 has been identified as a contaminant in IVIG, which can lead to thromboembolic events in patients receiving high-dose IVIG. So for this screen, the same load and conditions were used as previously. An equilibration buffer matching the pH and conductivity of the feedstock, and again, feedstock loaded, washed, and eluted. Several hits were identified, but 3 ligands in particular, F1, G1, and E8, were of further interest, which gave over 85% factor 11 removal with little to no binding of IgG. So, to verify the results of this library screen and to demonstrate the comparability of the MMM plate columns to more traditional columns, adsorbent G1 was selected. A 10-fold scale-up to a 2.5 ml borosilicate column was carried out, loading 20 ml of plasma. Elution was carried out using an equilibration buffer with the addition of 1 molar salt. This resulted in a classical affinity type purification profile with the target plasma proteins passing through the column unhindered and a small but discrete elution peak observed. Samples were analyzed by nephelometry, which is a dedicated analytical system used to determine the concentration of plasma proteins, as well as SDS page and ELISA for factor 11. Analysis of the collected fractions demonstrated good recovery of all tested plasma proteins. And using IgG as a performance indicator, greater than 90% of the IgG was recovered in the flow-through fraction. Adsorbent G1 was found to bind 94% of the factor XI present, which was subsequently recovered in the elution fraction, along with the binding and recovery of trace levels of other proteins. Process optimization. So a hit's been identified. In certain instances, this may be sufficient to move the hit to tech transfer and manufacture. However, in many cases, improvements to the adsorbent or the process may be required. This is also true for legacy products, where an older, previously good enough process now requires modifications to become more cost efficient or to improve their purity profile. In this case, process optimization can be investigated. In these following slides, I will focus more on the different aspects of downstream process optimization with case studies identifying different aspects of the process of the optimization process. Right. So ligand optimization. This is where we look, this is the chemistry side of it, where we look at the ligand density. We'll look at low and high to determine the binding performance and to get good binding capacities. We'll look at the spacer arms for the attachment of the ligands to the base bead. We'll look at the bed support itself. Prometic has 3 different beads for pure bead-based materials, which have subtly different properties. We'll also look at the chemistry of attachment and the adsorbent synthesis optimization. This is where we're trying to make sure that the materials that we use are suitable, that the process is cost-effective. In downstream process optimization, we'll look at verification runs. So this is looking at when the chemistry department produce the different ligand densities, spacer arm, bed support materials, we'll carry out verification runs to make sure that the process is working. We'll carry out binding capacity studies as well as wash studies to look for removing any problematic non-target materials. We'll carry out elution studies and CIP studies to improve the reusability of the materials as well. And finally, we also look at buffer studies. And this is where we want to ensure that there's no constraints with certain buffers to the proteins and to make sure that we keep the process as simple, as effective as possible. Kind of what the slide is also showing is in the first image where you see the binding and elution, we're getting a single elution peak which is not as pure, and we're moving that with wash and elution conditions to identify a sharp, discrete elution condition with high purity. PBL routinely will carry out design of experiments, or DOE, as a tool to rapidly identify multiple factors for process optimization. In this case study, the ligand density along with loading optimization was investigated. After an initial screen, a DOE study was carried out using a full central composite design, looking at 3 factors: salt from 300 millimolar to 1 molar, residence time from 1 minute to 10 minutes, and ligand density. Several adsorbents were made using the ligand optimization, and 3 adsorbents with the ligand densities of 12 micromole per gram, 15 micromole per gram, and 18 micromole per gram were selected for this study. For this target protein, investigating pH was not an option, as the protein was pH sensitive. The parameters of binding capacity, recovery, and purity were evaluated. So here we have some classic designs from the DOE, where we're showing from high binding or high recoveries in red and lower in the blues and the greens. The results of the DOE showed an improved binding capacity and elution recovery was attained when the ligand density was increased. However, in this case, increasing the ligand density led to a decrease in the purity of the target protein in the elution, i.e. we saw increased non-specific binding. Increased elution purity was obtained by increasing the salt concentration in the equilibration buffer, with only a small decrease of about 10% drop in binding capacity. And as expected, increasing the residence time led to increasing binding capacities in the presence of the increased salt concentration. So the original conditions which we looked at was a citrate buffer at pH 6 containing half molar salt with a 5-minute residence time, and the ligand density was 16 micromole per gram. The optimized conditions identified was the same citrate buffer at pH 6, which now included a 1 molar salt addition with a 10-minute residence time. Again, for the optimized conditions, the ligand density remained at 16 micromole per gram. I guess we'd already serendipitously picked the most suitable density in the original adsorbent. In this next case study, PBL collaborated with Albumetics. They developed several albumin fusion proteins, again, to increase the circulatory half-life of functionally active proteins. This collaboration was to develop a novel, selective, high-capacity adsorbent to bind albumin as a platform for the purification of albumin fusion proteins. Binding and purification studies of a single-chain FD fusion protein from a fermentation supernatant was carried out. The breakthrough study, carried out by Albumetics, shows that the flow rate of loading the feedstock, or changing the residence time, affects the binding capacity, as we saw before. It's also identified that along with changing the flow rates, lowering the pH of the feedstock from pH 6 down to pH 4.5, which in this case was feasible, improved target binding. The binding capacity, which was determined by HPLC, was improved approximately twofold from around 20 mg per ml to nearly 40 mg per ml. Further studies included a wash study of another albumin fusion protein, IL-1RA. To improve the purity of the target, the wash study investigated various buffers to remove non-specifically bound non-target proteins, which identified wash steps. The identified wash steps were successive pH increments at pH 6, 7, and 8, so seeing the removal of non-target material and improve the purity of the final target, as seen in lanes 8 and 9 of the STS page. Although from the STS page there is target material visible in the wash, I'd like to just have a closer look of the dilutions carried out, highlighting that only a small amount of target was lost in the wash steps, which were loaded on neat, and the majority was recovered in the elution fraction, which was a 1 in 100 or 1 in 1,000 dilution. So the implementation of AlbuPure for the purification of albumin fusion proteins over the existing standard ion exchange process not only improved the yield by about 50% with comparable host cell protein levels, but this also allowed for a process step reduction from 3-step to a 2-step process, which will decrease the production costs as well. Right. I've spoken about loading and wash studies. So in this case, I'd like to discuss a little about elution optimization. In this case, we purified 5 fragments from an E. coli lysate. The starting conditions was a neutral equilibration buffer, followed by loading filtered E. coli lysate onto packed absorbent F1P HF column. Post-load wash was carried out with the equilibration buffer, and the fag fragments were eluted with a pH 3 sodium citrate buffer. And as you can see on the FDS page, the elution contained fag fragments, however, requires significant optimization to remove many non-target impurities. An initial buffer screen was carried out. an elution of Fab fragments was observed at the lower pH range with increased non-specific protein elution at pH 9. A secondary screen was carried out, narrowing the pH range from pH 5 down to pH 3 with various buffer solutions, as well as including pH 9 to investigate that wash depth. The screen identified that milder acidic conditions between pH 4 and pH 5 gave significantly improved purity of the target protein compared to the lower pH conditions. And citrate buffer was observed as being more selective than the acetate buffer. pH 9 demonstrated good clearance of non-target protein. However, we also saw, in this case, increased levels of Fab fragments. Verification chromatography was carried out with the optimized elution condition, which was sodium citrate pH 4.5. From the starting condition, eluting at pH 3, as shown with the previous slide as well, and shown with column run 1, the elution fraction contained Fab fragments with a host of other non-target proteins. The optimized elution condition, using a citrate buffer at pH 4.5, demonstrated a vastly improved purity of Fab fragments as seen in lane 11, with minimal losses of target material in the pH 3 strip, which is in lane 12. In another study, we investigated the optimization of a clean-in-place, or CIP, using a caustic-sensitive adsorbent. A range of non-caustic CIP conditions, including chaotrophs and denaturants, were used at various concentrations. The SDS page here shows that residual material bound to the adsorbent. Higher concentrations of guanidine, as seen in lane 9 of the first gel, and acetic acid, conveniently as seen in lane 9 of the second gel, improved the cleaning of the adsorbent, allowing for improved reusability. So all cases up to now discussed provide a picture into the types of process optimization that we carry out. Once the adsorbent and the process has been fully identified, the process moves to the next phase. PBL have a dedicated tech transfer team which ensures the smooth handover of methods from R&D to manufacture. This includes not only adsorbent manufacture, but the transfer of all associated analytical methods, including assay validation, as well as column packing methods. Batch manufacturing records, or BMRs, are generated, and if required, further training is carried out for any changes to methods, new methods, or processes. In place, there is a routine preventative maintenance and calibration programs, change control systems, as well as line clearance to ensure that there's no cross-contamination. Prometic has a dedicated facility on the Isle of Man for the manufacture of adsorbents in ISO 8 environments and column packing in ISO 7 environments, which are continually cleaned and monitored, providing the highest standards of products to the customer. Prometic adhered to a quality management system with full documentation and procedures, with validation programs for all adsorbents as well as for column packing if that's required. Prometic can provide discrete 1,000-liter batches of material with the capability of supplying over 37,000 liters of adsorbent per annum. And to date, PBL have 18 products which are or have been used in FDA or EMEA licensed products and have packed our adsorbents in some of the largest radial and axial columns around. All of our procedures and equipment have been designed to provide a scalable platform producing products to GMP standards for our customers compatible for use in manufacture. So, on to the offerings that we supply to our customers. Whatever your requirements, PBL can supply, whether it's a range of off-the-shelf adsorbents and resins, including general purification adsorbents, as well as affinity adsorbents for the purification of insulin, albumin, antibody fragments, or contamination removal of serine proteases, isoagglutinins, and endotoxin. Of course, as mentioned in the presentation, we carry out many client-based programs as well, generating specific affinity adsorbents for our customers' needs. PBL also provides column hardware from MMM plates and small research columns from 1 mil kits, Evolve R 5 and 50 mil columns, to pilot or process Evolve and Evolve D columns, which are an open platform for PBL or custom adsorbents and are packed and packaged ready for manufacture. And of course, PBL are not just stopping there. We are currently evolving our column range, looking at making larger Evolve D columns with a 25 centimeter diameter, as well as smaller GMP-ready columns to support the growing needs with the emergence of individual gene therapy treatments. In closing, to support the growing need for more cost-effective, improved, and tailored therapies, PBL are providing a turnkey solution, from ligand discovery through process optimization with seamless TT to a range of manufactured selective separation products. Through our technology, our people, and processes, we provide the capability to enable biotech and biotherapeutic companies. Thank you for your time and attention. Please let me know if you have any questions, and we'll be happy to answer them.
Thank you, Buzz, for an excellent presentation. We've received a few questions already, but we'll give the rest of you a moment to enter your questions into the Q&A box to the left-hand of the slides. Before we begin with the Q&A, I'll run through some brief announcements. First, I'd like to thank Prometic Bioseparations for sponsoring this event. Next, I'd like to draw yourself quickly to the face-to-face BPI conferences visiting Boston this year and Kyoto, Amsterdam, and Santa Clara next year. At these leading annual bioprocessing events, you can hear from hundreds of expert speakers and network with peers. Bioprocess International Academy courses are developed and delivered by a global network of leading experts who have over 10 years hands-on industry experience and are skilled trainers, so you can expect a practical perspective and direct feedback on the challenges you are facing daily. Learn more by visiting the website at the bottom of this slide. Also, be sure to check out the resources list to the left of your screen, where you can download a large collection of useful resources, including a survey sponsored by Roche Custom Biotech, evaluating the need for a rapid sample-to-results benchtop microplasma testing system. If you or a colleague have experience with testing of microplasm, please complete the 5-minute survey. Now back to Chloe and Chris to begin the Q&A. Chloe, what is the success rate of the customer ligand development projects that you have worked on?
So our success rate, which is based on the achievement of the specification that sets with the client at the beginning of the project, is in the range of 80 to 90%.
Perfect, thank you. And Chris, can you work with the different base matrices, or is it just Purabead?
Well, the majority of our products in manufacture are based on Purabead, which is our proprietary agarose-based matrix. So this is beaded agarose. But we also have products in production based on polymethacrylate resin. And in R&D, we have experience with immobilizing ligands to polystyrene-based matrix, polymethacrylate, cellulose-based matrices. So yeah, we have a good experience with a range of different solid supports.
Thank you. And Chloe, how long does it take to screen one library?
So screening a library doesn't actually take that long. For the actual screening itself, you're probably looking in the region of 45 minutes to an hour and a half, depending on how many steps that you want in there. And that's when we're typically loading around 1 to 2 mils of feedstock. If you wanted or needed to load more feedstock, it will extend that time. Things that take a little bit longer is there's also the setup, and the longest part is usually the assays to analyse the fractions from the screen.
Thank you. Well, that looks like all the time we have today for questions. So thank you, Buzz, Chloe, and Chris for a great session. If anyone submitted a question that wasn't answered, keep in mind that the speaker will reach out to you directly. This session was recorded. You'll receive a notification in 24 hours when the on-demand session is available for viewing. Also, don't forget about all the downloadable resources in the resources box to the left of the slides. As mentioned before, Rush Custom Biotech has a survey on the need for a rapid sample-to-result benchtop microplasm testing system. So if you or a colleague are interested, please take their 5-minute survey. Before you log off, please take a moment to complete the feedback form so we can continue to improve your Digital Week experience. On behalf of Connect 365 Life Sciences, have a good day.